This is an AQA GCSE Biology paper 1H from June 22. Question 1. This question is about cells and transport. Complete table 1. So which part of the cell contains genetic information? It is the nucleus. Remember, that controls the activities of the cell. The genetic information is in the form of DNA. Mitochondria is the site of aerobic respiration. So that's where energy is released. Controls the movement of substances into and out of the cell is the cell membrane. Cells and potatoes are plant cells. Cells and potatoes do not contain chloroplast. What is the function of chloroplast? It's where photosynthesis takes place. Name one type of cell in a potato plant that does not contain chloroplast. So remember, chloroplasts require the action of sunlight in order for photosynthesis to take place. So think of a part of the plant which doesn't get any exposure to sunlight, and something therefore like a root hair cell is a good bet here. A student investigated the effect of salt concentration on pieces of potato. This should be screaming osmosis at you. This is the method used. Cut 3 pieces of potato of the same size, record the mass, add 150 centimetres cubed of 0.4 mole salt solution, place each potato piece into the beaker. After 30 minutes, remove each potato piece, dry the surface, record the mass, and then repeat using different concentrations of salt solution. What is the independent variable in this investigation? So that's what you change. Read the method carefully. That's my cat. We are using different concentrations of salt solution. Why did the student dry the surface of each potato piece with a paper towel in step 5? That's to remove any excess solution, which would add extra mass to our readings. The student calculated the percentage change in mass of each potato piece. For one potato piece, the starting mass was 2.5 grams, the end mass was 2.7. Calculate the percentage increase in mass using this equation. So, percentage increase equals the increase in mass, so work out the difference between those two numbers, over the starting mass, which we've been told is 2.5, times by 100. The final answer here is an 8% increase in mass. The student used the results from each potato piece to calculate the mean percentage change in mass at each concentration. Complete figure 1. You should label the x-axis, use a suitable scale, plot the data, draw a line of best fit. So let's start by labeling the x-axis. Just steal the wording here, and then you don't need to worry about units. Then let's look, we go from 0 to 0.4. That's 0.1. Make sure you're drawing along the line they've given you. Don't be tempted to draw any lower. So we've done that. Now we're plotting the data. Just so I can see it, I'm going to move it down. So 0 has a percentage change in mass of 9.8. 0.1 is 9.5. 0.2 is 7. 0.3 is 0.4. And 0.4 is minus 1.4. And now we want to draw a line of best fit. I'm going to struggle to do this neatly on my iPad. Oh, something like that for your curved line of best fit. So I've checked it all off to make sure I've got all the marks. What concentration of salt solution was equal to the concentration of solution inside the potato pieces? So this is where you, by definition, have a 0% change in mass. So effectively, you want to read along this axis. So it's that point there, which, read it carefully, is around 0.34. Explain why the potato pieces in the 0.4 mole salt solution decreased in mass. This is an answer relating to osmosis then. So which way will that water be moving? Well, water leaves the potato piece by osmosis. Water moves from an area of higher water concentration, and really specify where that is inside the potato, to an area of lower water concentration. Surrounding solution across a semi-permeable membrane. So be really specific with your answers here. Make sure you've got that definition of osmosis in there. You could have also written that in terms of water potential or dilute solutions, whatever wording you prefer. Plant cells and fungal cells are similar in structure. Figure 2 shows a fungal cell. Lyra, name one structure in figure 2 which is present in both plant cells and fungal cells but not in animal cells. Look, it's a vacuole. Remember, animal cells don't have that. You could have also said a cell wall. Which disease is caused by a fungus? Not gonorrhea. Malaria is caused by a protoctist. Measles is a virus. It is rose black spot. A fungal cell divides once every 90 minutes. How many times would this fungal cell divide in 24 hours? So let's get 24 hours into minutes by timesing by 60. So we have this many minutes in a day. And then how many times did it divide? Divide that number by 90. So it would have divided 16 times. Some types of fungal cell are grown to produce high protein food. The high protein food can be used to make meat-free burgers. Where is protein digested in the human digestive system? Stomach. It's definitely not large intestine or liver. They're trying to trick you with salivary glands. Remember, that's amylase. Which chemical could be used to test if burgers contain protein? Benedict's reagent, no, because that tests for glucose. Biray is correct. Ethanol is used to test for fats. Iodine is used to test for starch. Table 3 shows some information about burgers made from meat and meat-free burgers. Evaluate the use of burgers made from meat compared with meat-free burgers in providing humans with a healthy, balanced diet. Use information from Table 3 and your own knowledge. Burgers made from meat contain more protein. And then let's write why that's a good thing. Protein is needed for growth and repair of muscle cells. So we've done that one. What about fibre? Okay, we've got more fibre in the meat-free burgers. Meat-free burgers contain more fibre. Let's say what fibre does. Fibre prevents constipation, so that's a good thing. Then we have fat. Burgers made from meat contain more fat. That's a good or bad thing, depending on how you look at it. Fat is needed for insulation. But remember, diets high in saturated fat can lead to coronary heart disease. So that's a nice point we've made there. Carbohydrates around the same, so I won't really talk about that. I suppose we could say that they both contain a good amount of carbohydrate which is needed to provide energy. And then last up, cholesterol. Wow, look at that. Burgers made from meat contain a huge amount of cholesterol. I'm just going to copy my wording here. And again, that's a bad thing because it contributes to an increased likelihood of getting coronary heart disease. You could then talk a little bit about the ethics, talking about the fact that the meat burgers obviously involve the slaughtering of animals, the fact that the growth of those cows, if you're making beef burgers, releases a lot of methane, which causes global warming. So you have lots of options, but I've already got my 6 marks. A student prepared some onion cells. The student viewed the onion cells using a light microscope. This is the method used. Cut an onion into pieces using a sharp knife. Peel off a thin layer of onion from one piece of onion. Place the onion epidermis onto a microscope slide in a single flat layer. Add 3 drops of iodine. Slowly lower a cover slip at an angle onto the onion epidermis. Place the slide on the stage. Table 4 shows a risk assessment. Iodine solution is an irritant. It may cause allergic reaction or skin rash. Plan to minimize risk. Let's just clean up any spills. Sharp knife, the risk is that you may cut yourself. So what should we do here? Cut away from the body. Or cut on a chopping board, anything sensible. Give a reason for each of the following steps in the method. Why are we using a thin layer of onion epidermis? To enable us to see individual cells. Iodine solution is added to the onion epidermis. It acts as a stain. The cover slip is lowered onto the onion epidermis at an angle. That's to prevent air bubbles being trapped. Figure 3 shows what the student saw under the microscope at a magnification of times 400. The length of the cell in figure 3 is 4.8. Calculate the real length, give your answer in micrometers. So this is our formula triangle. So we want the real length, which is the object size. So we do object size equals I, which is image size, over magnification. Let's get this into micrometers first of all. You want to times it by 10 to get it into millimeters, and then times it by 1,000 to get it into micrometers. So that's where a lot of your marks will be awarded, getting those units right. We've been told that it's times 400. Pop that into your calculator to get 120 micrometers as your final answer. Figure 4 shows the student's drawing of figure 3. Give 2 ways the student could improve the drawing shown in figure 4. Lots of options. You shouldn't really be shading in a biological diagram. Maybe draw all of the cells present. You could label it, so label the nuclei. You could include a magnification or scale. Onion cells can be seen using an electron microscope. Give 2 ways onion cells would look different when seen using an electron microscope. You'd have a higher resolution. You'd also be able to see them in 3D. You could have also said that they would look more magnified or have more detail. Plant and animals have many defense responses Table 5 shows some plant defences. Identify whether each defence is a chemical or physical response. Thick, waxy layer on leaf surface. Hopefully you can see because that's thick, that's physical. Berries that are poisonous, they're releasing a toxin, it's chemical. Bark that falls off the tree is again physical. Mimicry is a mechanical adaptation seen in both plants and animals. Figure 5 shows 2 insects. Hornets are insects that sting other animals and cause pain. Hornet moths do not sting other animals. Suggest how mimicry helps the hornet moth survive. So yeah, usually that moth would be at risk of being eaten by predators, but the fact that it looks like a hornet means that predators will be too scared to eat it because they'll be afraid that they'll get stung. Adult hornet moths lay eggs that hatch into larvae. Figure 6 shows the larvae of the hornet moth. The larvae of the hornet moth live inside the roots of trees, use the tree roots as a source of food, cause damage. Explain why a tree might die if the roots of the tree are damaged. Okay, so let's talk about what the roots do. They absorb water and mineral ions, and they also support the tree itself and stop it falling over in high winds. So, if tree roots are damaged, less water is absorbed. So why do plants need water? For photosynthesis. So less photosynthesis occurs. Meaning less glucose is made. And less cellulose needed for cell walls. If tree roots are damaged, less mineral ions are absorbed. Why is that a problem? Fewer nitrates mean fewer proteins are made. Fewer magnesium ions mean less chlorophyll and less photosynthesis. So you really need to know what these various ions do. And then just generally, damaged roots mean that the tree is anchored or supported less well. The larvae of the hornet moth form when fertilized eggs divide by mitosis. Describe how mitosis produces 2 genetically identical cells. You're going to need quite an in-depth answer here, so it's all the stages of mitosis. The first stage is that the chromosomes are copied, and that's so that the new cell has its own genetic material. And then remember, they line up at the equator of the cell before those chromosomes are pulled apart to opposite ends of the cell. To produce 2 new cells, you need that cytoplasm to divide into 2. Because those chromosomes have been copied, the new cells are genetically identical. The cells which are first formed from the fertilized eggs of the hornet moth are stem cells. Name the process by which these stem cells then form specialised cells. That's known as differentiation. Question 5. Water and CO2 are exchanged between leaves and the atmosphere through pores called stomata. Name the cells that control the opening and closing of the stomata. Those are the guard cells. Water moves through a plant in the transpiration stream. Describe two differences between the transpiration stream and translocation. Well, firstly, transpiration stream just involves the movement of water and mineral ions, whereas translocation involves the movement of sugars. There's not an awful lot of room here to write this. Then in terms of the direction, transpiration stream occurs in the xylem upwards. Whereas translocation takes place in the phloem and is in both directions. Which environmental conditions would cause the rate of transpiration to be the greatest? So you want it to be windy, you want it to be dry, you want it to be hot. So let's try and pick out this combination. Cold with low humidity, no, because cold is not hot. Cold with high humidity, no. Warm with low humidity, yeah, that's the same as saying dry and hot. Figure 7 shows information about the mean width of the stomata in a plant. The changes in the mean width of the stomata in normal conditions are an advantage to the plant. Explain how. So let's have a look. At midday, we can see that we have a nice peak there with the mean width of the stomata. So at midday, they're going to be letting in lots of CO2 in, which is needed for photosynthesis. And at midday, you'd expect there to be lots of light, again, corresponding to lots of photosynthesis. At midnight, there's no light, so no photosynthesis. They're going to choose to close those stomata in order to prevent excess water loss by transpiration. The changes in the mean width of the stomata in low atmospheric carbon dioxide are different from the changes in normal conditions. Explain how the difference helps the plant to survive in low atmospheric carbon dioxide. Have a look here on the graph. You can see that in the low atmospheric CO2, the stomata are open wider and for more time. So look, the peak's higher, that's wider, and that allows the plant to take in more carbon dioxide for photosynthesis. Table 6 shows information about 5 different organisms. Calculate value x in Table 6. Give your answer to the nearest whole number. So we're looking for the surface area to volume ratio. So we look at these two numbers, identify the smallest number, it's this one, and divide both of those numbers by that number. I'll show you what I mean. Because that's how you're going to get your ratio. So there we are, dividing both sides by the smallest number to get a ratio of 1 to... So that's 73.7 recurring, so giving our answer to the nearest whole number, that becomes 74 to 1. What is the relationship between the size of an organism and its surface area to volume ratio? So here's our largest volume organism. It has the smallest surface area to volume ratio compared with this tiny organism. So as size increases, surface area to volume ratio decreases. Organism B exchanges gases with the environment directly through its skin, so it must have a large surface area to volume ratio to do that. Organism D exchanges gases with the environment using its respiratory system, so that's something like us, an animal, human. So that must have a low surface area to volume ratio, because that's why we need a respiratory system, to increase the efficiency. Oops, I've accidentally already answered that. So organism B has a larger surface area to volume ratio compared to D. For organism B, diffusion is fast enough, because remember, diffusion rates increase with larger surface area to volume ratios, to supply oxygen and remove CO2, so that's gas exchange. Table 6 is repeated below. Table 7 shows information about organism D and E. There's some metabolic rates. Organisms D and E both keep a constant body temperature. They're warm-blooded. Explain why the metabolic rate of organism D is greater than the metabolic rate of organism E. That's worth 4 marks. So let's compare their surface area to volume ratios. So organism D has a large surface area to volume ratio, and that means it will lose heat far more quickly. So it needs a higher metabolic rate, because that's all to do with the rate at which chemical reactions take place in their body, which means it will be respiring faster in order to generate that heat. Organism D and E both have alveoli in the lungs and villi in the small intestine. Describe how alveoli and the villi are adapted to increase absorption. They both have a large surface area. Why is that important? To increase the speed of diffusion. So remember, from an alveoli point of view, diffusion will be occurring in terms of oxygen moving into the blood capillaries and CO2 diffusing out. The villi, we've got that large surface area, again increasing diffusion rates of those soluble food molecules. They've even got microvilli. We could then talk about the proximity or closeness of the blood capillaries to both the villi and the alveoli, providing a shorter diffusion distance. They have thin walls for the same reason. So they both have a large surface area to increase the rate of diffusion. Villi even have microvilli. They both have thin walls. To reduce the diffusion distance. They both have lots of blood capillaries to maintain the concentration gradient. That just basically means they maintain a steep concentration gradient, because those gases and food molecules get taken away quickly, which means that you have a low concentration area, so therefore diffusion can occur faster. HIV is a pathogen. Give one way HIV can spread from one person to another through sexual intercourse. Table 8 shows information about new cases of HIV diagnosed in the UK. Describe the trends shown in Table 8 between 2010 and 2018. Now for women, you can see from that data, the number decreases, then increases, then decreases again. The number of cases in men, it increases and then decreases. Suggest one reason for the change in the number of new HIV cases between 2014 and 2018. So for both, we see this huge decrease. So what would decrease the number of HIV cases? It could be better education on how it's spread. It could be that condoms are more widely available, which will help prevent the spread. Better drugs to treat their HIV infection. I'm going to go for better education. Calculate the ratio of new cases of HIV in women to new cases of HIV in men in 2018. Give your answer to 3 significant figures. So I've pulled those 2 relevant numbers from the table. This is the number of new cases of women, the number of new cases of men. Make sure you're doing it so that it's the ratio of new cases in women to new cases in men. It means that we want men to equal 1, which means we divide 242 by 1,288. So here's our number. And then they want it to 3 significant figures, so that becomes 0.188. In the UK population, the total number of women is greater than the total number of men. The data in Table 8 is used to compare the proportions of new cases of HIV in the population for men and women. Suggests how the data could be presented differently so a more valid comparison can be made. Ideally, you'd want to calculate the number of cases as a percentage, because then it doesn't really matter how many people you have of each gender. Scientists have been working to produce a vaccine for HIV for many years. Explain how a vaccine of HIV could work to prevent a person developing HIV infection. So let's talk about how a vaccine works. So weakened or dead HIV is injected into the patient. Remember that your body responds in the following way. The lymphocytes or white blood cells, depends what your teachers taught you or what textbook you've read, recognize the antigens on the HIV surface and produce specific antibodies. If the person is reinfected with HIV, the good thing about vaccines is that this time antibodies are produced much faster, sooner, and in greater quantity. These antibodies kill the HIV. A person with late-stage HIV infection has AIDS. Scientists have produced monoclonal antibodies for HIV. The monoclonal antibodies can prevent a person infected with HIV developing AIDS. Describe how the monoclonal antibody for HIV can be produced. So the first thing to do is to get that HIV into the mouse, because remember, the mouse has lymphocytes which will produce antibodies which are specific to that HIV. Then the lymphocytes are combined with a tumour cell to produce what's known as a hybridoma. That's a kind of hybrid cell which has the properties of being able to produce antibodies to kill the HIV, as well as being super fast replicating, as is the case with a tumour cell. And then that hybridoma is cloned in order to create huge numbers of copies of that antibody. So to begin with, HIV antigens are injected into a mouse. Poor mouse. Lymphocytes from the mouse are combined with a tumour cell to make a hybridoma cell. The hybridoma makes antibodies which can kill the HIV. This hybridoma is cloned so that large quantities of antibody can be made. There are different ways of wording this, by the way. The MOT seems quite flexible. Figure 9 shows how HIV enters a human cell. So here are the small protein molecules on the edge of that human cell, known as an antigen binding site. The HIV itself has small molecules called antigens on the side of it. You can see how that HIV antigen is complementary to this binding site. And then that HIV virus has injected its genetic material into our human cell, therefore infecting it. Suggests how the monoclonal antibody for HIV helps to prevent a person infected with HIV developing AIDS. Use information from figure 9. So the way in which that monoclonal antibody for HIV is going to work is that it's going to be complementary. The monoclonal antibody will be complementary to that HIV antigen, meaning that the HIV bonds to the monoclonal antibody instead of the human cell, and therefore that HIV can no longer infect the human cell.